Ladies and gentlemen, and I'd like to request Dr. Ankita Pandey from PETA to join us on stage in the meantime. Dr. Sanjay Kumar and Dr. Arup Ghosh is on his way. So, he's just there. Hi. Very well in time, sir. Yes, sir. Please. This session is basically on regulatory mechanisms for biological agri-inputs. So, I'd like to request Dr. Ankita Pandey from PETA. I believe you got to introduce yourself because I'm lost now.
Good morning, everyone. I am Dr. Ankita. I'm working as a science policy advisor with PETA India. At the very outset, I would like to thank Mr. Saini and BASAI for inviting me to speak today. I think it's a great privilege for me to be sharing this platform with such inspiring women, especially on National Science Day. It's wonderful to see how they've, carved a niche for themselves in this male-dominated world, and they're, striving towards excellence. So, congratulations to everybody on that. So, for all of those who are not familiar with PETA India, we are basically an animal rights organization. Our primary area of interest is animal protection, but it's also to promote good science. And we've been working very closely with several government regulatory agencies, with companies and with scientists to promote non-animal methods of research and testing. Before I begin, I would like to quickly introduce you to the PETA Science Consortium International. It's basically an international group of around 25 scientists from all over the world. That is 9 PETA entities, including India. And we've been very closely collaborating with several scientific and regulatory organizations throughout the world to advance the uptake of non-animal approaches. So some of the ways how we're able to achieve this is through funding. We fund the development and validation of new non-animal approaches. We organize a lot of trainings and workshops and webinars to create more awareness. We publish and present at conferences, and we also conduct a lot of retrospective reviews along with regulatory agencies that help shape guidance documents towards replacing and reducing the use of animals. So in my today's presentation, I would just be walking you through with what is wrong with the historical approach of toxicity testing that has been dependent on animals, why it's being currently challenged, what options do we have if not testing on animals. We'll also be discussing how regulatory agencies all around the world, they are changing their practices to align with these new technologies. What are the current challenges that we are facing with respect to the regulatory acceptance and implementation of these methods? And I'll just be, to conclude, I'll be sharing some insights into what could be useful future directions. So when we speak of toxicity assessment, these have largely remained based on a checklist of animal tests that have been traditionally been considered as a gold standard. But the issues with the scientific robustness of such methods, it's increasingly being evident in the last couple of decades. So taking a closer look at what the limitations of this historical approach is, animal tests, they are known to have poor reliability, poor reproducibility, they are cost and time intensive. They also have questionable human relevance owing to the differences between humans and animals with respect to anatomy, physiology, metabolism, etc. Meaning that the data from these animal tests may not always be protective of human health. We know that science, technology, it's constantly modernizing, it's evolving, and animal tests do not represent the most up-to-date science. In fact, many of these methods date back to more than hundreds of years, and some of these were never even formally validated. What's also important is that as we speak, there are thousands of chemicals that still remain untested. So we really need to have systems which are faster, which are more efficient, so that we can keep up with the increasing number of assessments that we need to bring in new products into the market. What's also concerning is that these tests are quite animal intensive. For example, more than 10,000 animals are used to test just a single pesticide active ingredient. Fortunately, the need to have more faster, reliable, fit-for-purpose, human-relevant methods, it's kind of led to a paradigm shift towards the development and use of non-animal approaches, or as we call them, new approach methodologies, which are faster, they're cheaper, and they're more definitely fit for purpose. So taking a closer look, these are some of the examples of widely used non-animal approaches which are internationally available. They are scientifically valid and recognized, and they're either being used as a standalone or they are being integrated together to make protective risk decisions. Due to paucity of time, I will not be delving into each of these, but if you are interested, you need more information, please feel free to connect with me after the presentation. So when we speak of in vitro, ex vivo approaches, these are available for assessment of a lot of endpoints such as skin irritation, eye irritation, and corrosion. So the beauty of these systems are that these are actually based on human-derived 3D reconstructed tissue models that allow for better understanding of human mechanisms of toxicity, meaning that the data from these models will actually be more predictive of human health and more reliable. For skin sensitization, for example, we have another battery of assays ranging from cell-based assays to chemical activity-based assessments, which are arranged in an adverse outcome pathway where every assay represents a key step that is involved in the skin sensitization process. Similarly, for ecotoxicity, we have the rainbow trout gill cell line assay that is replacing direct testing on fishes for assessment of acute toxicity. Moving on to computational models, these are also becoming increasingly popular among regulators and industry for toxicity assessment. A very popular method is the OECD QSR toolbox. This is nothing but quantitative structure-activity relationships where toxicity is predicted using the chemical structure and the physiochemical properties. Another example would be the CATMOS tool that is also used for predicting acute oral toxicity. If we move on to waivers, these are a very important component of non-animal approaches. So sometimes not all studies or data requirements may be appropriate for all category of products, and certain toxicity requirements can be waived off if it's accompanied by a sound scientific rationale. So it's basically asking the right kind of questions before you begin testing. Is the assessment of this endpoint necessary? Is it relevant? Is it adding any new information to our risk assessment process? So some of these waivers are usually based on, say, the physiochemical, biological properties, bridging data, technical feasibility, use and exposure patterns, and so on. So US EPA, the Environmental Protection Agency, is one of the agencies that is widely using this approach to give out waivers and to do away with unnecessary testing. And as per their published study from 2012 to 2018, such waivers have saved as much as 300 million in study costs, 6 million on study review costs, in more than 2 lakh animals. So what I'm really trying to say here is that by doing away with unnecessary amount of testing, which is not adding any value to your risk assessment process, you're not just saving animal lives here, but you're allowing that your time, your resources, your money to be dedicated towards more relevant form of testing. Yeah. If we take a look at the international landscape, we see regulatory agencies recognizing the importance of these non-animal approaches. In fact, the US EPA even has a work plan to phase out the use of animals without compromising protection to the human health and the environment. We've also seen a similar move from EFSA, the European Food Safety Authority, and more recently, Canada has also published its notice of intent to develop a strategy to replace and reduce the use of animals. Even industry, they're voicing similar opinions. They're even coming up with new frameworks where they can do away with animal testing and generate the required data using non-animal approaches. If we speak about India, substantial progress is being made when we talk about acceptance of non-animal approaches, even for biologics. So for biopesticides, we have the 2023 guidance document by CIBRC, which allows waivers to be accepted based on a weight of evidence approach. They will also be accepting data from alternative methods which do not use animals, as long as they are recognized by OECD and other regulatory authorities. Similarly, the draft guidelines for biostimulants, they say that animal testing needs to be avoided wherever it is possible, and that existing information may be used, waivers will be considered, and scientifically valid in vitro methods can also be used. Similarly, for nano-based agri-inputs, the guidelines recommend that before considering any new animal testing, you need to take into account existing information, the chemical and structural properties, data from any in vitro studies before you proceed for animal testing. While I say there is a lot of progress being made, there is still an opportunity to further transform the current toxicity paradigm and maximize the scientific benefits of this new technology. So the idea is to move away from an animal-centric checkbox approach towards 21st century weight of evidence risk assessment that's based on use and exposure patterns, examination of existing data, applying any non-testing strategies such as QSRs, read across, and if more data needs to be generated, then non-animal testing approaches should be used. An animal test should solely be considered only as a last resort. While I understand it's easier said than done, and there are a lot of translational barriers that exist, whether we talk from a regulatory acceptance point of view or from implementation. So, of course, there is a lot of historical inertia. We sometimes see a lot of apprehensions, even from the regulators, with respect to acceptance of non-animal approaches. There's definitely a lack of awareness on the availability of these alternatives. And even if people are aware, they do not have enough confidence to use them, or they may not know how to apply data from these methods. We also see a lot of methods which are accepted in other countries, but not yet accepted in India. There is also lack of clarity on the acceptance of specific non-animal methods, which may actually discourage the industry to submit data using these alternative methods. Again, not a lot of CROs are equipped with these new technologies, so accessibility, that poses a major challenge. I'm just winding up, Mr. Sen. Okay. So as a way forward, of course, we are working to create more awareness. We are presenting at conferences. We are having expert group sessions to discuss about these. We need more training, more education, strengthening of infrastructure capacity. For now, we just have guidelines accepting alternatives, but we really need the legislation to set a strong preference for using data on non-animal methods wherever they are available and applicable. There should be more international harmonization and regular engagement. There needs to be more scientific dialogue between the regulators and the industry. These could be either in the form of pre-submission consultations, where the industry gets to discuss and clarify their data needs before they can actually proceed with any form of testing. Just a moment to say thank you to BASAI, because they've been like a strong pillar of support, helping us promote this cause in India. Last year, we did a webinar series. We had experts from CIBRC, industry, and CRO. The link is here. If anybody's interested, please do have a look. Last year, we also did a session. We invited CIBRC, CROs, and industry to discuss how we can increase the rate of transition towards moving towards non-animal methods. Would just like to conclude by saying that replacing animal tests doesn't mean that we are halting scientific progress or putting humans or the environment at risk. Instead, implementing non-animal modern approaches will improve the quality as well as the humanity of a regulatory science. Thank you so much.
Thank you, Ankita. Really impressive talk. You need to join us back. I believe we have a plaque for you. Yeah, we do. So, ladies and gentlemen, I forgot to introduce her properly. You just heard to Dr. Ankita Pandey. She happens to be the Science Policy Advisor to PETA India and the PETA Science Consortium International. She has received a doctorate from CSIR, Indian Institute of Toxicological Research, in 2018, and is a recipient of the 2017 Tulsa Bai Educational Trust Award by the Indian Academy of Neurosciences, and has authored a book, Footprints of Toxicology in India. So if you ever have a chance, this book is worth reading. Thank you, Ankita, once again. Dr. Sanjay Kumar, an ex-regulator from the Secretariat of CIB and RC. He has been very instrumental in Efficacy, and now he runs his own consultancy and also is a consultant to the Indian Institute of Pesticide Formulation Technology, if I'm not wrong. So over to you, Dr. Sanjay, please.
Good morning, everybody. Myself, Dr. Sanjay Kumar. I worked in dietary plant protection for 34 years. And after taking voluntary retirement, I started working as a consultant. And now I am also working as a consultant with Institute of Pesticide Formulation Technology and to guide how the experiments to be conducted on bioefficacy evaluation. So now I am talking about the bioefficacy strategy requirement for chemical pesticides, biopesticides, and biostimulants. Because more or less the bioefficacy strategies are similar, except few parameters are different. So under the Insecticides Act, it is framed to regulate the import, manufacture, sale, transport with a view to prevent the risk to human beings or animals. Now in this, the term insecticide, it is coined during 68 when most of the pesticides were only insecticides. There were no fungicides, herbicides like that. Later, it get incorporated, means any substance specified in the schedule. Whatever in the schedule, it is covered under the Insecticide Act. And the other substances, including fungicides and weedicides, they have also been covered under the term insecticide. And the preparation of the containing of many more substances, like the combination products. Now, there is a misinterpretation of the schedule. So, like these insecticides, now the number of pesticides are there, like insecticides, acaricides, nematicides, all are covered under the term insecticide under the Act, and commonly they are called the pesticides. Now, there is a misconception about a substance which is not included in the schedule as on date. So, most people say, okay, we can freely market the product because it is not covered in the Insecticide Act. But the truth is that if any product is claimed to have any property of this insecticide, acaricide, nematicide, then the manufacturer has to request to CBRC for inclusion in the schedule before marketing and before getting registration. It is the prior requirement. Now another is the biostimulants, which are regulated under the FC orders. So basically biostimulants are referred to as to stimulate physiological process in plants. It means it is some different from the plant growth regulators. In plant growth regulators, they enhance or retard the growth of the plant. But here, this function is not there. Only they are stimulating the physiological process through the uptake of the nutrients from the soil or from the plant itself. Now the biostimulants now covers the botanical extract, protein, vitamin, like that. Here the biochemicals are also included. Now again there is a misconception about the biopesticides and biochemicals. In biopesticides, the biopesticide products contain the living microbes like bacteria, fungi, and viruses. Whereas in biochemicals, they contain fermentation products of microbials, like in the abamectin, amamectin, spinosad, and they are covered under the Insecticide Act. Whereas biochemicals are also appearing in the FCO. And they mentioned synthesized or produced by the metabolism of the living organisms. So just slight variation, how the biochemical will fall under the Insecticide Act, or they will fall under the FC order under the biostimulants. Now the bioefficacy data requirement for chemical pesticides as per CBSC guidelines, the bioefficacy phytost to season 3 location, natural enemies, it is required for insecticides only. Effect on soil beneficial microorganisms for seed treatment and soil applied pesticides. Now it is essential. Earlier it was not mentioned, but now it is mandatory to submit the soil beneficial microorganism data on seed treatment and soil applied pesticides, whether it is herbicide or whether it is insecticide. Effect on seed germination. For seed germination, only it is required for the seed treatment. But it is my opinion that better get for pre-emergence herbicides also, because they may also check on the germination of the crop. soil physiochemical for herbicides. Now calculation of benefit cost ratio. Earlier it was required only for combination products to say that how they are cost benefit when we are multiplying, when we are using the two pesticides. Now in the guidelines approved in 449th RC, they have mentioned it's for all products, whether it is a single or whether it is a combination products. After succeeding crop, it is on 3 crops for herbicides. Now there is another requirement, addition to seed test. Under chemistry, it is mentioned that it is required for insecticide. But it is my opinion that why only insecticide? Seeds are treated with fungicides also. So it should also be applicable to fungicides. So better to get the addition test for insecticide as well as for fungicide. Regarding persistence in plant, so RC has approved now in 421st RC that it is to be generated at 1x and 1.25 dose. It is approved by RC. Persistence in soil for 4 types of soil, persistence in water 3 types of soil. Now come to the residue in plant. Residue in plant, as CABRC has approved long back, it was for X and 2X dose. And it was still continuing to some extent. But when the committee was formed for requirement the persistent in plant, they approved only for persistence in plant 1x and 1.25 dose. But CIBRC now also asking for residue also. I have discussed many times, sir, it is not approved by CIBRC. Please get a notification, approval of RC, so that instead of 2x dose, residue 1x and 1.2x may be generated. They have not considered my request. On one meeting, they informed me that now onwards you have to generate data on 1x and 1.25x, not on 2x. I told them, sir, it is a verbal commitment. How we will like that it is required? Then it was told, what is discussed during this meeting, you send a mail to all concerns so that you have a written proof that it is discussed and you have a paper in your hand that it is to be required. So it is the data requirement for insecticide, fungicide, it is for 1 season and 4 location, herbicide 2 season, 3 location. For seed treatment, now they have mentioned 1 season at 3 locations only. And for seed treatment, one point of interest is there, कि MRL fixation is exempted. But in case the crop is consumed well before 30 days, like vegetables, spinach, coriander, etc., then MRL is required. For soil application, 1 season residue data is required. But if at harvest, the residue is found more than LOQ, then 2nd season data will also be required. For non-commercial, non-edible crops like jute, copper, rubber, they have exempted on residue and persistence in plant. It is not required as per 449 RC. Now, Bioefficacy data requirement for biopesticides and consortium of biopesticides. Bioefficacy factors is same, 2 season at minimum 3 agroparameters like for chemical pesticides. Lab BASAI, it is the requirement of chemistry. They mentioned dual culture. Dual culture actually it is for entopathogenic bacteria, or not for entopathogenic bacteria or viruses, only for antagonistic bacteria, fungi. Disease severity, root colonization for antagonistic bacteria and fungi. But since it is a requirement of chemistry division, if a biopesticide is claimed like Trichoderma hirgenium only for nematode control, they still insist that no, as per guidelines, dual culture data is required. It is replied, discussed, sir, it is a nematode, dual culture experiment cannot be conducted. They told no, as per requirement we have to be required. After all, what is done? In petri dish, nematode eggs were kept, fungus were kept, nematode egg viability was tested, and the report was submitted. And it is non-scientific. It is not dual culture. It is just to comply that your study has completed. So it should be requirement of the bioefficacy, not the chemistry. Which do not understand where, for which product, for which disease pest, this dual culture is required. Laboratory nutrition under bioefficacy, LC50 is required from the approved laboratories institutes. Effect on natural enemies for entomopathogenic bacteria and fungi only. Effect on soil beneficial microorganisms, it is not required. But it is better to get for seed treatment and soil applied biopesticides, as it is required for chemical pesticides. Anytime it can be asked. So better to get the data when we are treating the seeds. Effect on seed germination, it is not required, but better to get for seed treatment also. Okay, what is the percentage of seed growing? Additions of seed test, again it is better to get biopesticide also. Now time is running short, I just go faster. Now for biostimulants, they have mentioned the bioefficacy data is to be required on active ingredients, not due to nutrient content. Means what are the studies are going on? Simply they are going to like chemical, biological pesticides, the studies are going on. But it is clearly mentioned in the draft guidelines that the efficacy of biostimulants should be due to other active ingredients, not due to its nutrient content. Therefore, the agronomic efficacy trials, appropriate treatments, comparison of biostimulants with the equivalent concentration of micronutrients present in the final product should be included to prove that the beneficial effect is independent of its nutrient content. Suppose we have taken some plant product, the nutrient contents are also come. So it is to take into consideration that when we are generating the bioefficacy data, this concept is to be kept in protocol so that it can be fulfilled. No fortification is required. Yes. Reporting requirements. One is the when we report the data. So for reporting data, for when the population is there, sucking pest, borer pest like that, so population data is required. Or for fruit or pod infestation percent, this data is also required for pod damage and fruit infestation. Percent square balls local damage in case of cotton, it is required. Percent dead herds in case of maize crop, sugarcane, wheat, for the stem borer, top borer, like that is required. Percent leaf damage in case of leaf folder, like that is data required. Disease, weeds for density and dry wet is data required. It is also mentioned in the bioefficacy guidelines that there should be more than 70% reduction is required. So it means if suppose at last observation the percent reduction is coming to be 60% or 65%, it will not be considered. So it is advised better to get one more observation. So naturally it may be go to the beyond 70%. If it is no so, then apply one more spray. Then you will qualify your bioefficacy trials. Otherwise, it will not be accepted because the percent reduction is coming to the level 70%. So it is my advice. Reporting parameters. Thank you. The websites are available on the website. You can see it. Right? Thank you.
Thank you, Doc. I would have loved to hear you more, but I believe we're pressing on time.
One thing more.
Yeah, okay.
As I told that regarding the residue study X and 1.25X, I have discussed with and what mail I sent to them, please see. Where they say it is the written document with you.
Ladies and gentlemen, we have a couple of recorded presentations from overseas. We will begin with one and then again we'll come on a physical presentation. So we have a recorded presentation from Giel Tettelaar, who happens to be the chairperson of Global Network Group and the Stitching EMCI Register. two cutting-edge providers of certification across the globe. Giel has a dual background in certification compliance as well as software development, and this background means that his certification bodies have an industry-leading certification process that is efficient, online, automated, and dynamic. Giel Tettelaar couldn't be with us today, but on September 9th this year, later on, we will be having a special session with him, and we will send separate invitations on this regard. Ladies and gentlemen, the presentation of
I'm the
Giel.
chairman of the EFCI register. We are one of the appointed notified bodies for the EU fertilizing products regulation. And in today's presentation, I want to give you a short overview of what the new fertilizing products regulation is and what it could potentially mean for companies that want to export their product to the European Union and want to use the FPR as a way of doing so. Just to start with a short introduction, the Fertilizing Products Regulation, or FPR 2019-1009, as it's sometimes called, creates a single harmonized market for EU fertilizing products. It's important to note that it is optional. It does not replace national rules. It instead creates a different pathway of marketing your products. And so if you already sell, for example, fertilizers or biostimulants to the European Union, you can continue to do so. However, with this new pathway, the FPR creates a way of legally selling a large range of new types of fertilizing products. These include fertilizers, organic fertilizers, inorganic fertilizers, and organo-mineral fertilizers, but also more novel products like biostimulants. It does that by creating a few different ways of doing what's called conformity assessment. These are divided up into modules. A lot of products can be self-certified, which means you can assess them yourself. You do not need to involve a regulatory authority or a notified body. And simply by performing an internal assessment, you can market the product in the EU. Some products, like for example biostimulants, require the involvement of what's called a notified body. This is a private conformity assessment body that can issue you a certificate and indicate to regulatory authorities, to customers, as well as internally that you do indeed comply. There are two procedures for bringing biostimulants onto the market. This is the module B plus C and the module D1. Under module B plus C, you perform a design assessment of a specific product. We would assess that the product itself complies. This would all happen remotely. We would assess that the product is effective. And once the product complies, we would issue the certificate. There's also the option to do module D1 assessment. That involves physical inspection and an assessment of the company's management system. Now, the way the FPR works is it creates categories for different types of components that are used to make fertilizing products. And depending on the components that specific products fall under, they have to comply with specific requirements. Some materials, for example, those where composting is used as a processing method, require the management system assessment. Others don't. The FPR is the first time at a European level that there is a single framework for biostimulants. There is a method for selling them, and there is a method to prove their efficacy. And indeed, notified bodies are required for biostimulants. And what the FPR also introduces is the principle of CE marking, which is a guarantee that when your product complies, you can sell the product in every single EU country without additional national rules. And to follow up on those national rules, what's important is that there are stark differences between the new EU regulation that went into effect 2 years ago and the national rules that are very common in today's stimulant market. Firstly, there is a single registration. You do one assessment procedure with a single body, notified body, and instead of having to go through 27 different countries or doing mutual recognition, you have a single certificate that shows that you comply for every single country. That also means that there are significantly lower administrative burdens if you can indeed fall under the FPR, but it also means that there are clear rules for selling throughout the European Union. There are strict requirements surrounding efficacy, and trials do have to be performed. Having said that, especially when managing different registrations, in the long term, the FPR potentially is cheaper. But it is also a lot faster to get to market because the assessment timelines with notified bodies are relatively short. Assessments are usually done within a matter of a few weeks instead of months or years, as used in other regulatory schemes within the European Union. And what is important to note, that even though you can continue to sell a product under national rules, some national authorities are implementing the FPR as a requirement and potentially even a replacement for their national rules. So when we speak of getting to market in the FPR, we will talk of conformity assessment and certification. And in that regard, these notified bodies are very important because they essentially replace registration authorities. You do not need to deal with national public authorities because you have to deal with private certification bodies. That also means you choose your notified body. It does not matter which one you choose. You just have to choose one, and the one that you work with, once you have a certificate from them, you can get to market. That also means notified bodies are pragmatic. You can approach them, they will answer your questions, and they will support. Once you submit a dossier to a notified body and you get approved, you get a certificate with a certificate number, and that allows you to get to market. It is as simple as that. And certification is valid for a period of 3 to 5 years, which also means you have some predictability in how you can continue to sell the product in future. Because changes must also be reported by the manufacturer, there is a lot of responsibility on the manufacturer, not just to report changes, but also to make sure that they continue to sell a compliant product. So you have to take this obligation seriously. Of course, when someone speaks of a regulatory regime within the European Union becoming more efficient and potentially even easier or more straightforward to comply with, the question is always, what is the catch? And in truth, there is not really one. Certification is in principle straight to the point. There are a few documents that you need. These are things like your analysis of nutrients and contaminants and pathogens in the product. You need to understand what are the ingredients in your product and how do they comply. You provide an explanation of the claims of your product. What does the product do? And that you have to prove. And of course, you have to prepare a label. In principle, the timeline is a matter of a few weeks. Then you get an assessment report. If the product complies, it can also be sent to, it can also be certified. That also means your lead time is relatively short, and it also keeps the cost low. In principle, the range for certification is between the 2,500 euros and 4,000 euros, depending on the complexity of your claims. Under Module B, usually there is a 5-year expiry date. And if after 5 years your product does not change and it still complies because the regulation itself hasn't changed, then there's also no recertification fee. So as a short overview, how does this process work? Firstly, and this is an example of one notified body, specifically the EFCI register, how we would approach it. And we would always start by trying to understand what is your product. And so we would set up an initial Teams call to understand what is your situation, what are you making, and what is the best plan of action. We would also make an offer for a fixed price, so you know upfront what you have to expect. You would sign a contract with a notified body after filling in an application form. And after that, that's the first step to be completed. And now there are only 2 more steps to go. In step 2, you would send in information, documents, other information on your product, files, and we would assess it. Any non-conformities that are raised, you could resolve, and you would resolve them by sending in more information. Once everything is compliant, you would complete step 2. Within step 3, we would review the information, we would perform a few administrative actions, and if everything complies, we would issue the certificate. In principle, you can decide the timeline. If you send in everything quickly and everything is prepared, it is really a matter of a few weeks. If these things take more time, then potentially you have to wait a few more weeks than the regular timeline, because of course you also potentially need more information from, for example, suppliers, which might take time to gather. It's important to note that non-conformities are not a problem. They're both an opportunity to improve the product or the technical documentation, but more importantly than that, they also do not lead to a denial of certification. The process will simply be stopped until the non-conformities are removed. And so it is something you can correct, you can implement fixes for, and you can proceed towards getting your certificate and getting the product to market. And so what does it actually look like when we're looking at a certificate? Well, it looks very much like this. It's always a specifically defined format for your specific product. It identifies you, it identifies the product, it has claims on the label relating to the specific biostimulant claims that are proven, and it can be verified. It comes with a certificate number that you can use, and then you can sell the product throughout the EU. That's it. This is all that you need. If you have this, a biostimulant can be sold in the EU in every single country. And then some frequently asked questions, and this is by no means an exhaustive list. So if there are any questions that have arisen, either during this talk or just in general that you might have from selling to the EU, do please feel free to always reach out. But as a starting point, it is important to note that you do not always need trials, but you do always need evidence. And if you do not have specific field trials on your product, then potentially literature studies are an option. Either way, the field trial standard, the field trials have to comply with a specific European technical specification. You can search for this online and you can download a copy, but there are specific requirements on things like how many replicates and what the plot size is for the trials that you perform. A notified body or an EU consultant can review your field trial designs, and indeed we would recommend this to make sure that you comply with the standard and make sure that there are no design issues in trials that you do perform. Indeed, the EU regulation also speaks of having to do a risk assessment. And what is key here is that you have an understanding of what your product is and what the risks are that are associated with this. If you have a safety data sheet, you understand that the type of product that you're selling or that you're applying for certification has been used before. In practice, you don't really need to do a risk assessment. But if it's very novel, potentially it's something that's worth considering. The FPR, when you dive into it, also contains a list of microorganisms. Specifically, it contains 4 strains. Any microorganisms which are not included in that list or in that those 4 families cannot be sold yet. These are Azospirillum, Mycorrhiza, Rhizobium, and Azotobacter. Any other strains, any other species cannot yet be sold under the FPR regulation. Animal byproducts, anything relating to that can only be sold when the delegated act, so when the regulation is amended. At the moment, it is not yet possible. When we look at technical documentation, it is also important to note that there are quite a few requirements surrounding analysis and testing. And it is important that the testing is performed in an EU laboratory that works under accreditation, which means if you are preparing for assessing a file or submitting a file under the FPR, make sure you have, at least for the notified body, EU accredited analysis. Of course, the methods for analysis are really up to you. The manufacturer can choose which methods are most suitable for their product, where the EU methods are preferred. But if you can prove equivalence to national methods, that is fine. And the choice is with the manufacturer. It is also important to note when it comes to the efficacy trials themselves that you have to define claims and you have to prove them. But you don't necessarily have to start with all of your claims that you want for the product and prove them. If you want to start with a smaller set of claims and then expand that list of claims later on for your label, you can do this by simply submitting in new field trial reports. And as said earlier, you don't need to work with a specific national authority. You work with a notified body, they can explain to you what the specific requirements are, and then from there on, you can submit your file and get a certificate. Once you have a certificate, you can get to market. That's the big new change in approach the FPR introduces. And as a summary, I would say it's impossible to make a complete overview of this regulation and the full impact that it has on you as an individual manufacturer. This is a brief overview of the most important points, namely that trials are required, that this is a new route to market, and that it does create a pathway for biostimulants. But if you want specific input on your situation, always feel free to reach out. There are no obligations in that regard. If you just have some questions, feel free to send us an email, and we will be more than happy to help and provide input on how the EU regulation could potentially work for you, or perhaps not. With that, I want to thank you all for the time for listening. Unfortunately, I couldn't be there, but I hope next year that can be different, and I wish you all a very successful conference.
Ladies and gentlemen, our next speaker is none other but Dr. Arup Ghosh. I'm sure all of you are familiar of him. Dr. Arup Ghosh is a senior principal scientist at the CSIR Central Salt and Marine Chemicals Research Institute in Bhavnagar, and is also a professor at the Academy of Science and Innovative Research in Ghaziabad. He is a renowned scientist, one of the authorities we believe in seaweed. And sir, I hand it over to you. Please do it just, yeah, it's already there.
Thank you very much, Bipinji. I thank you very much for giving me an opportunity to speak in front of all of you, the august audience. Basically, I am from CSMCRI. It's a research institute. This is a CSIR Research Institute in Bhavnagar, Gujarat. And we work in many things that are related to seawater. So anything that is related to seawater that revolves around it. My specific topic for today would be seaweed biostimulants, brief about what are the bioactives, what are the mechanism of action, their effect on crop growth, quality, and something about the regulations also. So to start with, let me give a brief overview of what CSMCRI is about. So we work on marine products and processes, marine biology, marine environment. In marine products, we deal with salts, we deal with water, marine chemicals, both inorganic and organic, marine elements, and marine biology, we do seaweeds, microalgae, halophytes, plants which are only growing in high salinity condition, salt tolerant plants, and we try to develop even genetically modified plants that have salt tolerance property, marine microbes, marine bioactives, and different products, valorization of seaweeds and the microbial products. We also have experts who have great expertise in assessing marine pollution, EIA studies, and impact assessment studies. So that is in a nutshell what we do. And you can see that we are a CSIR institute, and technology is our main focus area. So technology we develop, and we have been, considerably doing good on different things like composite of patents, publication, societal work, and visibility. So we are in fact ahead to some of the very reputed institutes on these counts. And this is a bit older data, but the recent data is also the same. And since I told that we are working with the industry, so we work with several different industries. We try to take the problems and then sort it out by different groups of scientists. We have about 90 scientists put together, apart from technical and other stuff. So that was in nutshell what we do at CSMCRI. Now I would be switching over to seaweed, biostimulants, and seaweed cultivation. You can see this is how seaweeds are cultivated. The productivity is much higher than any of the terrestrial plants per square meter area or whatever. The beauty is that it does not require arable lands, it does not require fertilizer, it does not require pesticides. So we as an institute have developed different technologies for cultivation, different types of seeds. You can see some are green, brown and red seaweed, which is a basic classification about. And we try to develop commercially viable value added products like agar, agarose, alginate, carrageenan and several other innovations out of these materials from seaweeds. So this is cultivation that is getting in Tamil Nadu coast. And this cultivation has actually helped a lot to the farming community, the coastal fishermen. Pattern of usage of income has increased, the lives of the woman that has in fact changed, in fact the numbers that accrue that are very important. Okay, so not all the methods are same for seaweed cultivation. It will depend on different coasts of India, because India is a huge coast. Not all the cultivation methodologies will be same. So this one is the earlier one I showed, raft method, that is suitable for Tamil Nadu. This one is suitable for places where there is high tidal current. So this is called tube net method, and that is how this seaweed, this is Gracilaria, Dura and Capophycus that has been cultivated in Gujarat. A very recent development is we have been executing a project wherein pan-India mapping of what are the conducive areas for seaweed cultivation has been initiated, and I think in another 6 months or so we'll be having an entire map of India where seaweed cultivation is possible, and that will be actually even there'll be there's a report, that is a policy, seaweed policy that is in the offing, and that will be very soon maybe released by our Niti Aayog. Coming to the technology, this the latest technology what CSMCRI has developed is a animal feed additive that is good for lactating cows, growing calves, egg layers and broilers. We have done this in collaboration with different ICR institutes like Central Avian Research Institute and National Dairy Research Institute, and we have toxicity trials also done. A good thing about is it can also reduce methane emissions from the ruminant animals, improve the body weight gain of the growing calves, and definitely improves immunity and the productivity like egg production and meat production of chicken. Coming to the biostimulants, world over there are only few species which are utilized for making biostimulants. You can see Ascophyllum nodosum, Eclonia maxima, Durvilia species. These are most of the important ones. Even 90% of the biostimulant market which we have in India is totally based on Ascophyllum. But in India also we have different species which are growing, which can be grown like Capophycus, Gracilaria, Sargassum, Turbinaria, and we are basically working on the Indian seaweeds, both their valorization, characterization. One thing is important, that composition vary with different types of seaweeds. So this is something very important from regulation point of view. You can see like I am comparing only two of them, like one is your red seaweed and another is a brown seaweed, both found in Indian coast, Capophycus and Sargassum. And we see that there are several things like total flavonoids, ascorbic acid, phenols, antioxidant, different species of polyphenolic compounds that are also very different. But that does not mean that there is a change in efficacy, because there are some things which are not found in one sort of seaweed, and others may have some other bioactives. Even we have done a comparison also between the ascophyllum and the native ones, and we found that it's comparable, and in some cases it is much better. So regarding the efficacy trials, that is also part of a regulation. We have actually carried out 175 trials. The requirement is only 3 trials, but we had carried out 175 trials. For example, in rice only, you can see we have carried out more than 20-30 trials all over the country by collaborating with different agriculture universities and ICR institutes. And with what Dr. Sanjay Kumar just now told, that it has to have a very specific statistical design. There are doses, like 3 doses. In these trials, we have done at least 4 doses for each of the seaweed biostimulant. There has to be proper replication, there has to be proper design, and then you find that we have found about 11 to 37% improvement in yield over and above the recommended doses of practices. We have done for maize, green gram, black gram. Again, the problem for regulation is like for crop labeling, we have to have trials for each of the crops. But we saw that in EU there is a provision that on 9 crops, if you do, it is applicable for all the crops, that is, I mean, in Europe. So some sort of thing that will be definitely required. It has to be discussed with the regulators, that how this can be done. For us, we did like 2 for cereals, 2 for pulses, 2 for oil seeds, 2 for sugar crops, like that. But I mean, the clarity is not there on this front. Not only on Carpa ficus, we have also done for Gracilaria, that is another seaweed that is found in Indian coasts, and same number of 175 trials for agronomic trials and another 165 to 70 trials. These are big scale demonstration trials on farmers' field. Similarly for sargassum, that is also we have done. This is not only good for plant, but it is also good for microalgae. Now this is the regulations, like what are the heavy metal contents. Even they have put the micronutrient contents, which are actually not very harmful. I mean, they are beneficial, but still they have put the limits, which is again different from EU regulations, because you can have that micronutrient included as a different and there are methods how you can do it, which here we cannot do. So these are the limits, and we can see that. See, I mean, now I'm just taking one example. We have done for many of the seaweed extracts. This is for only one extract. You see that there are a lot of constituents, including metals, heavy metals. For example, none of the heavy metals present in the seaweed sap will exceed the one that has been recommended by FCO. That we can always do. There are so many other compounds, organic compounds, bioactive compounds, you can see in the chart, belonging to several categories like sugars, sugar alcohols, amino acids, lipids, organic acids, amine derivatives, nitrogen compound. But do you want to test all of them? If you give this list to the regulators, are these bioactive compounds? For that also we have seen. See, these are the bioactive compounds that is found in Carpa ficus, but none of the regulators will have any method also to a standard method to actually evaluate them or even detect them. But are they bioactive at the physiological level? Yes, they have. I mean, even if you add one compound, synthetic compound, exogenously, that is found in literature, we have found that our seaweed biostimulants contain a lot many concoction of compounds which are in mixture. So they are not one, they are not two, there are many of them that is present together, which even exogenously, if it is applied, there are available literature that it gives effect. Okay, at the applied concentration I'm talking about, not the ingredient. But again, having said so, can we give? No. So there has to be some. These are the bioactive compounds, we cannot give them. So the suggested is like they want markers. So marker compounds, seaweeds will have markers, polysaccharides are the markers. Okay, so these are some of the suggestions which we have time and again given. Okay, like carrageenan, look for carrageenan, look for alginate, look for fucoidans. So these are some things which we have discussed. I mean, it depends again on the regulators what they are going to do, but yeah, definitely there should be, and I mean, not all the compounds are going to be present all the time, because the time of harvest is important, the season of harvest is important, even the stage of the harvest is important. Okay, so I am not going to put so many things, like we have done research, photosynthesis improved, nutrient content improved, nitrogen content especially, the whole lot of transcriptome work we have established, how without having any significant nitrogen compound, why it is important, why the nitrogen content is increasing. That is because there are two genes that are upregulated, that is nitrate reductase and the nitrogen transporters present in the roots. So that is what we have done, and we have published, I think, more than 50 research papers, especially targeting each of the mechanism, each of the scientific mechanism. I can share the presentation with Vipinji, so you can have a look at all the publication of whatever is your interest. So I'm not going to go into the detail, and that's a very good, disease resistance mechanism, how it is important for soil microbes. This is, for example, soil microbes, how it is important. Even under drought condition, it is going to increase the diversity and population of the soil microbes. This we have done through metagenomics, 16S rRNA, next genome sequencing. And this is also good for, from policy point of view, this is my last slide. Policy point of view also, it is important because this is an extremely low carbon footprint product. We have calculated about, I think, 118.6 kg carbon dioxide equivalent per kiloliter of the production of biostimulant at industry level. This is the industry data, and we have published in several of the research papers. So ultimately, when you use low carbon footprint product by reducing at least 25% of the recommended dose of inorganic fertilizers, the carbon footprint per unit of production is much more reduced. So this, thank you. That is all I think for today. Thank you very much.
Thank you, Doc. Thank you, Doc. Wonderful presentation. I'm sorry, we are just running out of time, and we have a lot of speakers in place at this point of time. Ladies and gentlemen, I'll just come back. We have our next recorded presentation from Elisabeth Douce, who happens to be the Global Regulatory Lead Biosolutions at NPP by UPL. Her presentation is entitled Global Regulatory Landscape, New Emerging Areas for Biocontrol and Biostimulants.
I'm the Global Regulatory Lead for Natural Plant Protection, the UPL Biosolutions branch. NPP is a UPL OpenAg global brand dedicated to biosolutions. As all brands, we act with a single mindset and culture and a shared belief in advancing sustainable growth for all. I'm going to speak about new emerging areas for biocontrol and biostimulants on a global regulatory point of view. First of all, I would like to present you our NPP Biosolution portfolio, manufacturing sites and labs. Then we will deal with regulations for biocontrol, highlighting recent progress, more specifically about microbial cell-free extracts. Finally, we will discuss about fertilizer regulations, focusing on the EU harmonized regulation and the concept of product function categories. NPP ranks number 1 globally in biosolutions with 9 manufacturing sites and 5 R&D labs worldwide. NPP manufacturing sites are located in India, France, Mexico, and South Africa. R&D labs are located in India, France, Mexico, South Africa, and in the USA. 7 experimental centers allow us to test our solutions in the field from early stage projects. Here are some pictures of our manufacturing facilities and of our new brand research center in Mexico that opened last November. NPP product range covers all biosolutions. It consists in biocontrol, bionematicide, bioinsecticides, including bioacaricide and biogrubicide, as well as biofungicides. It also consists in biostimulants, nutrition, and inoculants. A huge diversity of product types is present in our portfolio, from botanicals to microbials, natural extracts, purified or not, as well as minerals, humic, and fulvic acid. Let's consider biocontrol regulatory topic. Within biocontrol, 3 categories are usually regulated as plant protection products: microbial, pheromones, and natural extracts. Brazil has legislation for microbiological products since 2006, and last year, in 2023 revision, the Brazilian government added the definition and requirements for registration of products based on inactivated microorganisms. Brazil now considers living but also inactivated microorganisms with specific data requirements. In the USA, microbial pesticide category also covers both living and inactivated cells. In the EU, microbial data requirements are designed for living-based microorganisms. Inactivated microorganisms go under natural extracts, which fall under general requirements. In India, under the Insecticide Act, only living microorganisms are considered. The Brazilian case is quite interesting. Indeed, Brazil introduced a simplified registration procedure for microbiological products, the Joint Ordinance of 2023. The registration of technical products is not required. Only the formulated product requires registration. In total, it lasts around 10 to 12 months from dossier submission till product registration. This ordinance covers both living and cell-free microorganisms. This last category corresponds to fermentation broth, where microorganisms have been inactivated intentionally by the manufacturer. Benefits are multiple. The natural complex mixture brings the required efficacy, it solves stability issues and brings longer shelf life. Let's discover fertilizer and biostimulant regulatory topic. Fertilizer regulations are very fragmented over the globe. Each single country raised specific rules, most often covering organic, organomineral, and inorganic fertilizers. Subcategories are defined with parameters that are usually fixed, such as the minimum amount for fertilizer element and organic carbon content. For fertilizer, many countries offer a direct go-to-market as far as the product complies with existing categories. In Brazil, the biostimulant effect of microorganism-based product is covered by the new joint 2023 ordinance for plant protection products, not by fertilizer legislation. Biostimulant registration implies dossier evaluation by authorities, as in EU, South Africa, and India. Let's check EU situation in more details. In the EU, the journey to harmonize regulation covering all types of fertilizers, including biostimulant, took more than 10 years. Regulation 2019-1009, published in 2019, became applicable in July 2022. Fertilizing product definition is very broad, with purpose of providing plants or mushrooms with nutrients or improving their nutrition efficiency. It's based on product function categories and component material categories. The EU fertilizing products correspond to a fertilizing product which is CE marked when made available on the market. The CE marking is applicable in all the 27 member states. 7 product function categories covers all possible functions related to plant nutrition and nutrition efficiency improvement. The PFCs have been defined as follows. Product function categories are described into 7 categories, the last offering the possibility to mix other PFC together. Within fertilizer, the organomineral category also gives the possibility to mix inorganic fertilizer, like NPK, and organic carbon-based components of biological origin. Soil improvers can be either organic or inorganic. Their function is to maintain chemical properties, the structure or the biological activity of the soil. In the field of biocontrol, regulatory rules are more advanced for microbial-based Brazil offers a complete framework for both living and inactivated microbes. In the field of fertilizers, most of the countries cover 3 types of organic fertilizers. The EU concept of product function category a much broader offer with a total of 7 PFCs, including organic fertilizers, but also organic and inorganic soil improvers. It gives the possibility to mix several PFCs. Across categories open more possibilities for agriculture, such as farmers. This is how we see the future of biocontrol and thanks. Please forward any question either to me or my colleague Ravi in the slide. Thanks a lot. Bye.
That was, ladies and gentlemen, Elisabeth from France, NPB by UPL and especially my UPL colleagues. Now, as we call an end to this, Sanjay Kumar, the senior, with all years going across, flowing around. So if you could sign of appreciation to Dr. And another one to Dr. Ankita Pandey. Ma'am, I'll request you to. And I'll ask, give it to Dr. Sanjay.